8.3. These are short too. So for number 1, what did we just say? What 2 molecules make up the backbone of DNA? We just took those notes. A nucleotide is made up of 3 things. 2 of those things make up the backbone. Good, sugar and phosphate. Yep, repeating units. Sugar, phosphate, sugar, phosphate, sugar, phosphate. And then what molecules make up the steps? If you're holding the ladder, which part are you stepping on? The A, T, C, and G, the nitrogen bases. Good. And what are the base pairing rules? What goes with what? Which letters stick with each other? Anyone else want to answer? We just went over it in our poll. Good, A with T and C with G. Yep, the stick letters stick together, the T and the A, and then the curly letters, the C and the G stick together. And what does it mean to replicate something? What's another word for replicate? Good, Desiree, yep, make a copy. So this section is about DNA replication using those base pairing rules. So essential question, what happens during the process of DNA replication? DNA replication copies genetic information in the nucleus of a cell. So this is the S phase. This is what happens in the S phase of the cell cycle. Way back when we talked about mitosis, we talked about the cell cycle. We had the G1 phase, S, G2, and M. M was mitosis. We had a whole chapter on that. S was DNA synthesis, when the DNA gets copied. And this section is about that phase. So step 1, there's 3 steps. First step, enzymes unzip or unwind the double helix to make a replication bubble. The enzyme that does this is called helicase. So if you remember, sugars end in OSE, like deoxyribose. Proteins, enzymes, end in ASE. We're going to talk about 3 different enzymes, and they're all going to end in ASE. So each strand acts as a template or hard copy. If you've ever made a copy of anything in a copy machine, you have that original paper that you put into the copy machine and then it gets copied. That original paper is called a template or a hard copy. Step 2, bases that are floating free in the nucleus bond to their mates in the original DNA strand. The enzyme that directs this part is called DNA polymerase. So for example, you can see in the bottom, if the parent has a T, the dotted strand is going to have what to pair with it? What goes with T? Make sure you know the answer to that. Step 3, it all gets glued together. And it gets glued together by enzymes. The enzyme in this case is called DNA ligase. And in the end, each new DNA has one old strand and one new strand in its double helix. DNA is the template, but enzymes do the actual work. DNA just kind of sits there and lets itself be copied. It doesn't really do anything. Enzymes do all the heavy lifting. And this process is called semi-conservative. Remember when we talked about semi-permeable, semi means part or half. We call this semi-conservative because each new DNA molecule has half old information and half new information. It saves our cells valuable energy not to have to recopy full DNA strands every time. We only have to copy half of it.